Today we're going to be hearing from Hani Goodarzi, which he started his lab, has been doing wonderful work for the last decade at UCSF and is now at the ARC Institute, right next door to where I am in Palo Alto, and is doing really cool work thinking about RNA structure, non-coding RNA. His lab has been integral in thinking about how non-coding RNA is involved in cancers, thinking about foundation models for RNA sequence. And today I'm very excited that we're going to get to hear about some of the newer work thinking about context-free grammar encodings for RNA and how this can help us understand RNA-protein interactions. So very excited. We're going to have questions at the end, so please put your questions in the chat or raise your hand at the end of the talk. All right, all yours.
Awesome. Thank you so much, Rachel. And thank you everyone for joining. Yeah, please keep the questions for the last. Some of them hopefully I'll answer as we go along, and we'll tackle what remains at the end. As Rachel mentioned, I'm Hani. I'm an associate professor at UCSF. I started my lab in 2016. I then moved to ARC Institute 2.5 years ago as a core investigator. I was also a founding member of the Computational Technology Center here at ARC. My own background, I entered biology from the dry side, really developing machine learning tools for biology. And today I'm going to just kind of start from the very beginning to motivate why we are interested in this problem and how we have been approaching it over the years, and how kind of responding to advances in the field, we have continued to look at how we can build RNA models that are really built from the ground up for modeling RNA. Great. So I really don't know kind of the background of the field, the audience. So I'll try to kind of be in the kind of lay side a little bit in the beginning, but we'll get more technical. These are my disclosures, nothing that would relate to the presentation today. So as this audience probably knows, we have a singular genome. All the cells in our body have the same genome, but of course they manifest this genome in different ways. And this is of course through a kind of central dogma and gene expression. And the idea is that there's a regulatory program in place that kind of governs this process and governs the flow of information from DNA to RNA and to protein, and that ultimately decides the cell fate, the cellular fate, and cellular function. Now, as Rachel mentioned, my lab is interested in biology of cancer and cancer progression specifically, and we also view this as fundamentally a disease of disordered gene expression. And the idea being that these regulatory programs and networks in the cell are often disrupted or hijacked by the disease processes, kind of during disease progression, to kind of evolve more and more metastatic and advanced forms of the disease. And this is often reflected in gene expression data from even matched. So what the data I'm showing on the right is really matched paired primary and mets from breast cancer patients. You see there's like fundamentally hundreds, if not thousands, of genes that are changing as the disease becomes metastatic. So there's kind of very clearly this large-scale reprogramming of the cancer genome that's happening, and the underlying regulatory programs involved. And we have, for the longest time, been interested in kind of understanding what are these regulatory programs that are hijacking these processes, and kind of coming up with de novo machine learning tools that would tell us de novo, kind of originating from the data, the identity of these pathways. Now, when we talk about gene expression, this is really oftentimes it really viewed as kind of this steady state RNA level, right? So this is when you say gene expression is really measured by either, traditionally microarrays and then more recently RNA sequencing over the past, 15 years or so. But it's really like RNA is kind of has been the focal point of a lot of our gene expression analysis and tools. Of course, we are seeing more and more work on the proteomic side and other measurements as well, but gene expression is fundamentally, the bulk of it has been at the RNA level. But of course, behind the scenes, there's a lot that is happening. when we talk about gene expression, from transcription, processing, export, localization, translation, and degradation, all of these things, even when we talk about RNAs, like we're talking about this, all of these things kind of combined. And of course, each of these steps are involved in the flow of information. Each of these steps are very well regulated in the cell, and disruptions in them associated with a variety of complex diseases, including cancer. And my lab has dedicated a lot of its time kind of understanding how we can actually do this. Okay, oh, sorry, just a second. Sorry, there was a meeting that was supposed to be canceled, wasn't. Sorry about the interruption. So when we talk about, again, the steady state mRNA levels, these are governed by both rate of transcription and rate of decay and degradation. And the idea is that, there's, of course, both cis-regulatory elements on DNA and RNA, and trans factors like transcription factors, chromatin modulators are on the RNA side, RNA-binding proteins and microRNAs that bind and interact with a DNA or RNA to kind of drive these regulatory processes. But what we are trying to understand is that kind of given the gene expression data, kind of, or really measurements of other measurements of RNA, how do we go back to identifying, kind of reverse engineering the identity of these regulatory modules that are involved either on the cis side or trans side? So this, of course, has been a long-standing problem, really from the first paper that introduced microarrays, also talked about cis regulatory elements, of course, initially in DNA and how we can find these elements. The idea behind it has always been that genes that are part of the shared regulon, they are co-expressed and they behave the same way or in similar ways, and therefore they share regulatory elements, in their promoters in case of transcription, or somewhere along the UTRs in case of post-transcriptional control. And we can actually, via measurements of RNA across conditions or in a few conditions, we can actually get a sense of what these elements could be. And really going back decades, there have been tools that have been kind of designed to do this. And some of them are actually still in use to date. By and large, they've, these have been very successful. We have found a number of, DNA motifs and RNA motifs that have stood the test of time. But in general, they're also associated with high false positive rates. Now, when I was a graduate student at Princeton in Said Tawazui's lab, I worked very closely with Olivier Lamento when then he was a postdoc at Princeton. Now he's the director of Englander Institute at Weill Cornell. And he and another postdoc in Said's lab are working on a tool called FIRE. And I joined that team and I worked on this project for a few years as well. And the idea behind it was basically how can we kind of build a systematic approach for finding cis-regulatory elements in DNA or RNA. And the idea behind it was to really take advantage of mutual information as the most kind of systematic and generalizable kind of approach to measuring interactions and associations. Any non-random association really can be captured by mutual information. It's also generalizable in the sense that it applies to both discrete and continuous variables. So you can apply it to, for example, cluster indices from clustering data, or from log fold changes or things like that. Where mutual information comes in is that you're simply measuring the association between two variables. On one side, measurements of expression, again, either discrete or continuous. And the other side, a binary vector of presence or absence for your cis-regulatory element. So for example, here on the left, you have this blue box, which is basically a motif in the promoter sequence of these genes. This is, of course, the cartoon, but it really shows a clear enrichment in cluster 1. For example, genes that are in cluster 1 tend to carry this element. Similarly, for this light blue element on the right, you can see an enrichment towards higher like log ratio values, so towards the top. And this can be, as I mentioned, captured using mutual information. The idea being that, you know, if the two variables, basically presence or absence of the motif, is not informative at all about the expression patterns, basically the joint distribution equals the multiplication of the two. So this becomes 1, log of 1 is 0, and mutual information becomes 0. And as there are associations emerge in the data, this deviates from, this term deviates from 0, and you can actually have higher and higher mutual information values, and it's actually measured in bits, because, you know, it's information. Now, in practice, what we do is that in FIRE, we start with enumerating all possible k-mers, for example, all possible 7-mers. I'll talk a little bit about like why this is important, but again, you start from all possible 7-mers and use mutual information to rapidly kind of calculate the association between each of these k-mers and the input data, and you sort them based on this mutual information. Right? And then you start from the top. Mutual information is not a statistic itself. It's simply carry, you know, it's basically a unit of bits. So you have to convert it to a statistical value, and we do this using kind of, you know, jackknifing and shufflings and all that. But we do this only for the significant k-mers. And the idea is that you start from the top, the highest value. You do kind of a greedy optimization for by adding degeneracies into the k-mer. So you turn the k-mer into a regular expression, and you continue to make changes while mutual information increases. So here you see you start from this k-mer with a mutual information of 0.06, and you optimize it to 0.08, and this becomes kind of the sequence motif logo that that you're all, you know, often familiar with, and you can actually push this further even more into like a position weight matrix, which is kind of what motifs sometimes are shown as, and that's actually part of my work in Saeed's lab as well, to be able to do this transition. But regardless, you know, for most all intents and purposes, regular expressions are quite effective in capturing these elements. Now, what happens next is that when you go to the through the k-mers, you do realize that some of them are actually just very similar manifestations of the first one. Right? So we don't want to do all of this process again and again for these that are basically, for example, the second one is like one transfer of the top one. Right? So what FIRE does is actually uses conditional information to remove redundant k-mers. So you will only optimize and look at k-mers that provide new information given what came before. Right. So for example, here you will go to the third one because this third one is actually quite different from the first one that we optimized, and so on and so forth. So this is basically how you would go about basically finding cis-regular determinants in DNA or RNA. But the idea is that, well, all of this k-mers are basically naturally just about linear sequence, right? And while we did apply this to RNA, the reality is that we all even knew at the time that RNA structure is quite important, and even more so today. We know that we need to be able to capture the influence of RNA structure on RNA-mediated and regulatory interactions. And there's been plenty of evidence for this, but when it comes to protein-RNA interactions, for example, based on a number of large-scale data sets that folks have generated, you can go and try to build models, with a variety of tools and approaches to be able to predict whether a given sequence will be bound by a given RNA-binding protein or not. Right. So, for example, that's this plot on the left. You can see for some proteins we can do a pretty good job. For others we can't. For example, POM2 is actually known to be have a very precise motif, and it's a linear motif, and you can do a pretty good job of capturing that. Whereas for some other RNA-binding proteins, it's very like the sequence elements are just not very well captured. So there's something else going on, and actually A2B1 is one of the earliest RNA-binding proteins that I showed as a graduate student that has this structural context to its interactions. So these are the kind of influences that we want to be able to capture, right? So what are the, not just the sequence, but also what are the structural contexts that drive these regulatory interactions? So how do we go about doing this? For linear elements, I talked about how we can go from k-mers to regular expressions to position rate matrices. And this transition is really only about being able to like enumerate the search space, right? K-mers are a finite number of k-mers, so you can enumerate all of them, and then you can basically further do a local search to turn them into regular expressions or position rate matrices. So what is the equivalent of this for structural elements, right? Structural elements are, the way to look at them is basically, the same RNA sequence, but there's other constraints on top of it, right? That we often show in various ways, either like in 2D graphs or these bracket kind of representations. But the point is that you basically tie different parts of the sequence to each other, right? So how do we actually capture this? So of course, this has been, again, a long-standing problem, and one of the key solutions that was presented a long time ago, and I remember actually first reading this paper a long time ago, and that actually put me on this track, 30 years ago. The bottom line is that we can take advantage of context-free grammars and context-free language. So what is the idea, right? When, let's say we are in the regular language domain, and we are talking about like a string like UGAC, right? The way that you can really think about producing this sequence as a set of production rules that one comes after another as a grammar, you basically start from nothing, you add a C to the previous nothing, you add an A to the previous C, you add a G to the previous AC, and ultimately you add a U. As you can see, we just add step by step every nucleotide, right? So this would be just simply a linear element. So this is basically a k-mer. What I've only done, I've broken it up in 2 steps. Now, when it comes to a structural element, you have this sequence, and as I mentioned, is a structural entanglement that you want to be able to capture. So how do we do this using context-free grammars? So context-free grammars kind of have one-step memory. And they do this by having this entanglement on both sides that you'll see in practice. So let's say I want to recreate this structural element as a context-free grammar with a series of production rules. So again, I start with nothing. I add the A. So we're going to make the loop first. So I'll add an A and a G. So this is basically like a linear element. But then when I get to the stem, I add a U on one side and A on the other side. Right, so these U and A are always tied to each other, regardless of what comes in the middle. So this is a production rule that when you apply it to the previous one, you basically get the UGAA kind of part of the stem loop, and then you add the other one, and then you add the other one. So this is basically a context-free grammar. That when you run through this series of each of these is a production rule. So when you kind of link these production rules together, you end up with this representation of a structural element that captures both the sequence and a structure here. Now, of course, others in the field have also taken advantage of this idea. A long time ago, for example, RNA-Promo came out that uses stochastic context-free grammars to be able to find structural elements in RNA. And the way that we used it is basically very similar to how we approached things in FIRE, but now focusing on context-free grammars instead of linear elements. So again, in the linear elements, we have k-mers and regular expressions, and for structural elements, we're going to basically replace both of these with context-free grammars. And again, the way that you normally show structural elements is this sequence of structure combinations or these 2D graphs, or as I presented here, you can also represent them as context-free grammars, a series of production rules. Again, the sequence is completely optional, so you can actually have basically a context-free grammar that is all ends, right? So, but with clear structural information. And once you can have the representation via context-free grammar, that would be like basically your seed or motif, then you can easily scan sequences, and the problem turns into what we had before, like a binary vector of matches to context-free grammars, and whether you want to, and whether there's an association between this binary vector and your measurements that can again be evaluated using mutual information, as we covered earlier. So in practice, the way that we do this, again, similar to our enumeration of all k-mers, we actually started with a very large kind of repertoire of kind of context-free grammar seeds, and this was done in collaboration with Hamid Najafabadi, who was a graduate student also at the time at McGill, and now he's a faculty there, and we continue to collaborate to date. So we basically start with a random sampling of this space, a coarse grain sampling of this space of smaller structural elements, and these are all like terminal stem loops, so like very much limited exploration of structural space. But each of these elements basically has some sequence and some structural information, right? And basically your compute and complexity of your problem defines like how many of these you can actually process. And again, so these are basically equivalent of our k-mers. We will use mutual information to score them. And finally, we used conditional information to, again, put the ones that are similar into these bags. And each of these bags kind of represent a definition of a structural element. So for example, like if you look at a regular expression is a bag of k-mers, which are all allowed, right? So the same way a structural element, we define it as a family of similar context-free grammars that together define kind of the rules of a structural influence. So we had used this approach for in a number of studies over the years to kind of find regulatory elements that have this strongest structural components to them. And most recently, back in 2021, we found a cool interaction between SNRPO1 and a structured RNA that drives the splicing programs and kind of prometastatic splicing programs. But again, this is kind of a traditional approach to finding a structural elements, right, or RNA cis-regulatory elements in general. In reality, the world has changed. We are in the era of deep learning and more contexts going into building sequence to function models or sequence to sequence models. And fundamentally, the question becomes, are these models capable of understanding structure as well, right? So a lot of these models are taking advantage of convolutional neural networks. Convolutions are very similar at core, are very similar to position weight matrices, right? That's basically what they represent, which again, position weight matrices are in principle like linear beings. So they're not very good at capturing structure. While some foundation models are shown to understand structural relationships like through evolution, they're not really explicitly modeling structure. So this is kind of the problem that we wanted to solve. Like, how can we bring kind of this structure awareness to deep learning models that we are using in genomics so that we can actually apply them to kind of RNA-centric problems. So how did we solve this problem? So this was in collaboration. This was work done by a joint postdoc between me and Bo Wang's lab at Vector when Bo was there a long time ago. Basically, Mehran and I came up, and Bo came up with this strategy where we are basically trying to change the format of the problem in a way that we can solve with context-free grammars. So again, if you look at this stem loop structure, you have a loop here. Let's say the loop doesn't matter, so it's n and n. And then the first part of the stem is a CG, a U, so on and so forth. So each of these production rules are basically building this stem. As I mentioned, this, anything can go in the middle of this production rule. But let's say if I, in this kind of a specific example, if I fix the loop, you basically get, a C and G 3 nucleotides apart, and then an A and U 5 nucleotides apart, and then U and A 7 nucleotides apart, so on and so forth. So basically, each of these production rules becomes a base pair with a set distance apart. So this is very much reminiscent of dilated convolutions. So what we do is exactly this. We create these fixed kernels that are not trainable, and they basically recreate each of these production rules. So for example, this first one can be represented as this kernel, where you basically have a CG 3 nucleotides apart. So when you scan the sequence, this will fire whenever there is a CG 3 nucleotides apart. And so in practice, the way that it works is that you start from the beginning of the sequence with this kernel, and it starts going along the sequence. These are like partial matches, so it keeps going. And once it gets to the CAG, which is a perfect match, it basically will maximally fire, and you will see that here. Okay, so this kernel will be able to find a perfect match in this way. So now that was one of them. Of course, you can do this for every other one as well. So S1 to S5, each of them can be represented as a dilated, fixed dilated convolutional kernel. So this one is AU 5 nucleotides apart, UA 7 nucleotides apart, so on and so forth, right? So each of these production rules can be represented as a kernel. That's also not trainable, right? These are fixed. Now, what is important is that, again, if you want to find the sequence elements, this is a structural element, and you want to represent it in the convolutional paradigm, all you need to do is basically take these kernels, these convolutional filters, and see where they line them up and see where they all fire, and that where they all fire is basically where you find a match to this structural element. But of course, now we want to go the opposite direction, right? Of course, these are not the only possible fixed convolutional kernels. Actually, every possible base pair interaction and every possible distance becomes a kernel. So it becomes a very massive, multi-channel kind of input here, or rather output here from this convolutional process. Now, so you have the output of all of these production rules acting on your input sequence. And of course, again, the complexity of the problem kind of decides how far your receptive field would be here in terms of the furthest possible interaction between positions. And now the idea is that if this element is actually useful to the model's task, the model will be able to actually learn a relationship and a link between these convolutional kernels independently and put them together and say, this is what I want, and this would be the representation of this element. So we are giving access to, by going through these production rules, we're giving access to a structural understanding to the model that if it's useful to the model, it can actually take advantage of it to be able to basically sense and understand structural influences on the task that we are measuring. So let's see a little bit of this in practice. So basically, if we take sequences from RFAM and we just pass them through this fixed dilated layer. Again, there's no learning here. You're just basically going from a 4-channel input to a many-channel input output. And this many-channel output, we basically do a principal component analysis on it, and you get a 2D projection of the RNAs, and you see a very nice separation based on family. And RFAM is basically a structural family, so it actually makes sense. So again, this process actually maintains this structural information in the sequences. Now, based on this approach, we created an architecture that we internally call Pythia. It basically looks like this. You have 2 arms One arm is basically a ResNet architecture that acts on the sequence, one hot encoded sequence. And the other arm first passes things through these fixed dilated convolutional layers, and then is another sort of ResNet, and then they get concatenated, and this is what becomes, goes towards, via an MLP, goes to your supervised task. So this arm is basically what most models these traditionally used to look at RNA models. So any kind of arm can come here. Any kind of arm can come here. It's just a matter of whether the model is acting on the one hotted sequence or what comes out of the fixed dilations. And I'll come back to why this dual arm was important to us. So let's take a look at how it will work in practice. We basically took random sequences that were completely devoid of any kind of information, and half of them we put a context-free grammar of interest that we created. And so half of them carried this context-free grammar and half of them didn't. And again, the idea is that can the model actually learn this separation? It turns out that it can. And if you look at the embedding of the model, you can see a very nice separation between the presence and absence of the context-free grammar. So again, context-free grammar is this a structural being, and the model can access that structure. And of course, you can take it further and look at things like pseudoknots that are traditionally a bit hard to classify, and again, very structural problems. And here I'm comparing Pythia to other models that we have trained on this data. Again, in classification task, and again, you see that Pythia does quite well across the board, especially with AUPRC. There's a very significant advantage for Pythia to be able to kind of identify what is a pseudoknot and what is not. Again, it's not surprising to us because we have designed it to be able to find these kind of interactions, longer range interactions between nucleotides. Of course, in the meantime, there have also been a number of benchmarks, including Beacon in the RNA space, that have some structural tasks. So we have tested Pythia on these tasks as well. Like one of them is contact map prediction. You can see that Pythia does quite well in this space. We were mainly focusing on the kind of other supervised model like ResNets or CNNs or LSTM. But in this specific task, you can see it's better than other reported models as well. Distance matrix prediction is another structural task in Beacon. Again, Pythia does better than other supervised models. Single-site mutational impact, another benchmark. Again, Pythia does quite well and does better than other supervised models. And finally, when it comes to secondary structure prediction, Pythia really stands out. Again, we built it to be able to explicitly be able to model these interactions. So it works as we expect, right? So it's capable of actually learning these structural rules quite effectively. So where do we actually apply this? So we did all of this work to be able to look at RNA-protein interactions, as I mentioned earlier, and kind of the influence of secondary structure and its context on RNA-protein interactions and the rules. And where we were applying this is actually a data set that we generated in collaboration with Manuel Leonitti's group at BioHub. So big shout out to them. So they had this project at the time called, I think, OpenCell, that they were systematically tagging with a split neon green kind of much of the proteome in hex cells. And they were doing imaging on this and also some IP mass spec to look at protein-protein interactions. We realized that because we had access to this resource and also how good of a kind of tag this neon green, split neon green tag was for really effective pull down and clean pull downs, we actually leveraged this to do kind of a large scale CLIP data set on 60 something RNA binding proteins. And we actually made quite a few modifications to the traditional CLIP to make it even more efficient. And so we call this variant ecCLIP, which Dr. Lisa Fishent, when I was a postdoc in my lab, she worked on this protocol. And the output of this, as I mentioned, is very high quality data for 60 something RNA binding proteins. And you can see the number of peaks and number of binding sites and where they bind and all that. And again, CLIP is a nucleotide resolution protocol. So that's a nice thing about that. One of the first explorations we did was basically comparing the binding site similarities to protein sequence similarities. And there is some correlation, not a lot. So just overall RBP sequence doesn't necessarily drive what it binds to, which is, both interesting and not so unexpected. We have also done kind of comparisons with the ecCLIP dataset, which also is a large-scale CLIP dataset of RNA-binding proteins in other cell lines other than HEC. Ours was HEC cells. I think those are HepG2 and K562 cells. But regardless, we looked at a number of metrics, for example, like fraction of reads in peak. So ecCLIP actually gives you a much better distribution of reads around peaks and all that. So in terms of quality, it seems to be a high quality data set. We also did comparisons with bind and seq, which is in vitro binding data set for RNA binding proteins. This is of course limited to the RNA binding proteins that were shared between our data set and Encodes data set. So it's only a handful of them. But the results have been very consistent that in general, we actually do see a very good correlation between what we see in vivo for RNA binding proteins in ecCLIP and the in vitro bind and seq data, whereas this was actually generally absent in the original kind of ecCLIP data set. So HRMPL, as an example, on the left, you see there isn't much of a correlation between the in vivo scores and the bind and seq in vitro scores for ecCLIP, but we see a nice correlation with ecCLIP. So all of that to say, it's a pretty high quality data set, and it's something that we can go ahead and basically build models on. So we have built a number of models, as you can see here on the x-axis. I told you about Pythia, but we have opinions, but we are not opinionated. Ultimately, we are driven by the data. So we basically tested a variety of approaches for building models here. Pythia does quite well as a supervised model. There are some foundation model embeddings that do well here. I have a bit of mixed feelings about this because in the end of the day, these models have seen our test set, at least at the sequence level. So there's a little bit of leakage there. But regardless, Pythia really stands out as a supervised model in being able to learn the interactions between RNA binding proteins and the sequences that they bind. And now going back to our kind of dual arm Pythia, the reason that this dual arm was interesting to us was that now that we have a model that, say, for RNA binding protein X predicts whether there's binding event or not, it can then ask which of these two arms is the model relying on for that prediction. The idea being that you can, by deciding which of these arms the information goes through, you can decide whether that is influenced by structure or by sequence and to what extent. So we basically ablate each of these two arms and look at the ratio of the two to basically capture the influence of a structure. And we can do this across all RNA binding proteins and all of their binding sites. So you get a map like this, that going from like left to right. So left is basically sequence dominant, and right would be more structure dominant, right? And some of these are just not unexpected, like ADAR being here, for example, or on this end, there's quite a few very known kind of linear sequence binding elements. Now, but what is important is that almost every RNA binding protein has some instances in which the structure seems to be kind of driving the interaction, or vice versa. You can see there's a fair bit of variation around that mean. And we picked some of these RNA binding proteins, like 4 from this side and 1 from this side, to go deeper a little bit and kind of experimentally validate this. So the validation was done in a way that we took the basically reference binding site for each of these 5 RNA binding proteins. And then we created mutant variants of them that would either maintain the structure or disrupt the structure, or like disrupt everything. What we did was we put these sequences into a reporter, and we did basically CLIP for these 5 RNA binding proteins and looked at how the binding changes to relative to the reference for each of these variants. And as you can see, by and large, we see that for these binding sites, which were expected to be structured, basically structurally influenced, uh, keeping the structure is actually helpful. So mutations that maintain the structure end up maintain some of the binding, but you get more of a drop in mutations that will disrupt the structure. And if you kind of disrupt everything, you just get rid of binding in general. So this kind of very nice validation of kind of the model at large that these structural influences actually are can be validated experimentally as well. So to summarize, I showed you basically I told you about context-free grammars, why they were important to begin with, and then I told you about how we can use these fixed dilated convolutional kernels to basically capture this kind of context-free grammar-like dependencies that can help encode RNA secondary structure. We showed one implementation of it in Pythia, but the reality is that I think you can take these dilation layers and like add them to any kind of model that you want, right? You can even not do it dual arm. You can even go just from a one hot encoder to adding many channels. So your input becomes a massive multi-channel input as opposed to a separate sequence channel. So you can basically capture these dependencies in kind of any model architecture that you want. Why are these encoding? But I showed you how this dual arm interpretation would help us be able to look at kind of the influence of a structure on the decision of the model around RNA-protein interactions. Again, some of the RNAs that, some of the RBPs that we know are like sequence binders, they do show like influences, structural influences and vice versa. So there's more complexity to the RNA-protein interaction landscape than we would like to just easily gloss over as categorizing them in one or the other. So this work really took a village over many years, as you can imagine, both on the experimental and the computational side. Mehran developed Pythia, Lisa developed ecCLIP and did clips for some 30-something RNA-binding proteins. Heather was another postdoc in the lab who did the rest of them. Heather and Lisa worked on the kind of validation step that I showed you as well. Big shout out to all of our collaborators. Bo co-mentored Mehran with me and was basically deeply involved in this project from the beginning on Pythia. Big shout out to BioHub and Manuel Leonetti for the amazing resource that they've built, and also Hamid's group that actually did all the kind of comparison to bind and seek and encode clip that I showed you earlier. So I'm gonna stop here and happy to answer any questions you might have.
All right. Thank you very much for that wonderful talk. And so Isaac Vach asked, I apologize if I missed some details, but can you discuss further how the fixed dilated convolutions in Pythia are constructed? What data is necessary to actually construct these? And how well is the dilated convolution expected to generalize across contexts and different applications?
So I would say it's not data dependent, right? So I mean, I gave you an example of how you construct them. In reality, this is not the way you go. I was taking the opposite route. In reality, you will basically make every possible dilation kernel. So for example, like at 3 apart, you will have 4 different versions. That will be GC, AU, UA, and CG. And then you will go to 7, 5, 7, all of those. So basically, you have all variations of this, and each of them will give you a channel, right? So and the number of channels will depend on how many of these you build. And the longest one in the end decides on your receptive field, right? That is the maximum length that you can explicitly define an interaction in a stem, basically. So how many of these you can do is basically it adds computational complexity, right? Because you're adding these channels. So the model becomes parameterized more and more. So it depends on how much data you have. The more data you have, the longer you can go without overfitting. But this is not input dependent. It's a fixed, unlearnable layer, convolutional layer, that you can apply to any kind of input to go from a 4-channel input, basically a one hot encoded input, to a many channel kind of base pair interaction output. And then you can add that to any kind of model and any kind of problem, right? So I showed you kind of examples of, kind of injecting something yourself and predicting it, pseudoknots, all of these different benchmarks and in Beacon, for example, and we applied it to this RNA-binding protein interaction data set, of course, because that's where our interest is. But in general, any problem, to be honest, I think this is how you should do deep learning in the space of RNA, because we all know structural elements are important and structure influences RNA regulatory interactions, and kind of most architectures just really ignore that. And this is an approach that to really explicitly define structural rules for the model to be able to tap into.
All right, thank you. We have some questions on model training from Sylvie and Pierre. I'm just going to combine, essentially asking more details for how the model's trained, what is used as ground truth, and what is used as kind of inputs to train the model.
So in this case, you can see there you get a peak for every RNA-binding protein. We basically took that peak and created dinucleotide shuffled versions of each one. So there's basically a 2-class classification. You can also do multi-class classification that we have done as well. So yeah, I mean, depends. In this case, it's kind of one versus scramble classification, but you can do other variations of that as well. So yeah, it starts from the peaks detected in the ecCLIP data set and goes from there. And I should also mention that we basically like leave out a chromosome as a test set, and so there's all of that as well.
Okay, the next question is from Roma Nagle. She's asking about Pythia predicting binary classification RBPs. Is there a way to extend this towards calculating binding affinities or continuous values in general? If so, are you able to decouple epistatic interactions occurring with RNA structure that may affect affinity non-linearly?
Yeah, I mean, the short answer is you can do regressions. So, some of these benchmarks I showed you, like this one, for example, is classification, but some of them are regression. So, this is R2 score here for kind of the SSI uh, benchmark. This is also a continuous task here, distance matrix prediction as well. So the final head of Pythia, you can really add anything you want here. So it can be a regressor or a classification, or it can be also multi-class or even multitask. So that part of it can be really anything. As I mentioned, like what comes after this is completely up to you. Um, uh, what is important is that this operation kind of explicitly tells the model the base pairing rules that we care about and we know about. So it's basically we are giving the model this inductive bias that we know that there is base pairing possibilities between these bases in the sequence. And if that information is useful to the model then learns how to take advantage of it.
I have a kind of technical question about the kind of structural kernels you have. You said long range interactions are possible to be modeled. How long range is long range, and what is limiting that kind of length scale in the model?
Um, Again, the complexity of the model, because you're adding for each added receptive, like a step in the receptive field, you add 4 kernels, right? So depending on the complexity of the problem you have and the number of data points that you have, you can define that. Here we were looking at RNA-protein interactions. The peaks are short to begin with, so you can actually cover all of them. So that's not a huge problem. But if you are thinking about like how can I put this into like a Borzoi type model or an alpha genome type model, it would be that would be a different conversation, right? So that whether you have enough data to be able to increase the receptive field to that point, to that extent, or even if you, and more importantly, whether you want to or not, I think these are kind of problem by problem. You have to define those and see if the model is not over parameterized and can actually learn.
Right, one more question in the chat is there a specific model of binding being discovered? Um,
I don't think I understand the question. A specific kind of binding? I mean, the bottom line is it all comes down to a thing like this plot, right? So, well, a combination of this plot, which is a question of given sequence, how well can you predict binding? And here we are showing AUPRC, for example.
I mean, the
Uh,
question, actually, I mean, so if finally you found a model of bending like a small stem loop is as a structure, and maybe the small sequence motif located in the hairpin loop, something like that, have been discovered.
really good question. I think so. This level of interpretation has been hard in context of like RNA-protein interactions, right? So for this example, it was pretty easy, right? We injected something in, like we injected a context-free grammar, and you can actually see those production rules of that context-free grammar as being important for the model. But when it comes to this kinds of RNA protein interaction kind of data, you have to realize that, for example, if there is variation around the base pairing rules, right? Each fixed dilation is a fixed distance. It's a gapped filter, right? So if you say, for example, I need to have a stem, but I don't really care about the size of the loop, like all of those stem possibilities become important. So they all contribute. To be honest, I'm still thinking about how we can like take the kind of saliency of these kernels and turn them into kind of a more simplified visual understanding of the stem loop structures that it's finding. We are working on it, haven't made a lot of progress, as much progress as we would want. So that part is still a bit lacking as how do you like visualize any of this beyond just the interpretation step that I showed you. But at the same time, I think visualization often implies simplicity that might just not be there, right? So from what we see in these interactions, that you do quite well in predicting the interactions, but then you see a fair bit of variation in terms of what did this interaction means in terms of the influence of the structure on said interaction.
Thank you.